Hello everyone and welcome to FSMA and Water Activity. Today's presentation will be 20 minutes, followed by 10 minutes of Q&A with Dr. Brady Carter, Senior Research Scientist here at Meter Group. If you have a question for Dr. Carter, type it into the questions pane at any time during the webinar. We'll be keeping track of these to ask during the Q&A. Please don't be shy and submit those questions. Also, we'll send a link to the on-demand webinar and the slides as soon as they're available, so you can review anything you missed during this presentation. So, without further ado, I'll hand the microphone to Dr. Brady Carter.
Thank you very much and welcome everybody to today's virtual seminar where we're going to be discussing the role of water activity in the new Food Safety and Modernization Act. And hopefully this will be an opportunity to be able to answer questions for you if you've been wondering, does water activity still applicable as part of FSMA? What role does it play? How do I use it? Has anything changed from what I was doing before? And so hopefully we can go through and talk about those things individually. We're not going to spend a huge amount of time covering FSMA because obviously that's a huge topic and takes multiple trainings and seminars. But what we're going to focus in on is what can you expect in terms of how you will use water activity as you adjust and take on the new requirements as part of this new FSMA regulations as they go into place. So, as I said, today we're going to talk about water activity and the Food Safety and Modernization Act, more commonly referred to as FSMA. And many of you are probably already starting to try to figure out what is associated with FSMA, what does it entail, what do I need to do, what do I need to focus on, and all the challenges that are associated with that, trying to figure that out. And so hopefully what we can do today is maybe at least help with a piece of that in starting to help you understand what the role water activity will have in that and how you might be able to use it. So let's just start off with a quick overview of what is FSMA, or the Food Safety and Modernization Act. And so it was a sweeping reform of food safety laws, much more so than changes that have occurred in the food code over the past. These were much bigger changes to the overall process for how those people who make food will handle it and what things they need to do. And really the main shift was from going from a reactive system where recalls were a way to deal with problems when they came up. And a lot of the focus was on what happens to the product after it's made and released, to being more proactive and being able to try and identify things that could happen before they do and then try and prevent them. So switching from a reactive to a preventative type of system. And so what this kind of boils down to in terms of how do you operate on a day-to-day basis is that now the system is requiring that you as a food producer create a food safety system. And so what does the food safety system consist of? And basically what this is taking a look at your process, your processing, and analyzing it to determine where are the potential pitfalls and areas that you could have problems, and then how do you prevent that from happening. And so the steps include, first of all, doing a thorough hazard analysis. So this is, again, going in, looking at your processes and determining where is there a potential problem, where could something happen that would cause your product to go bad, or to potentially cause people to get sick, and to have that could result in a foodborne illness. And so you go through and look at each part of your process, from ingredient acceptance to your processing time, your kill step, to your packaging, to your handling afterwards. And you do an analysis of what the hazards are. And this is may not be a new thing to many of you, especially if you've used HACCP in the past for your safety programs. This is basically the same concept with a few differences as HACCP, where you looked at hazard analysis and then you identify critical control points. So again, here the same thing, hazard analysis. You're looking for risk. Where is your greatest risk? And so as you assess each of these hazards and you do a risk analysis or a risk determination, you're basically looking at two things. Number one, how likely is it the event is to happen? And number two, if the event happens, how serious is that going to be? How big of a problem is it going to be? And so through that, you can do a risk assessment, and from that risk assessment, identify the particular hazards that are of most concern. And then once you've determined that, then you can start to put in some preventative controls. So I've identified these hazards as my greatest risk. How am I going to prevent them from happening? And so then you identify different things that you can do to prevent those things from happening, not react to them when they do happen, but prevent them from happening. And so when you establish those preventative controls, along with that goes a necessity to monitor those preventative controls. So how are you going to make sure that those preventative controls are in place, they're at the proper level, and that comes through monitoring. And this is really where testing becomes very important, testing your product, QA, QC testing on the product to make sure that it's meeting the requirements for the preventative controls that you've established as part of your food safety program. And then, of course, it's important to build into the program a way to do corrective actions. So if something you identify through your monitoring preventative control that's not where it's supposed to be, what are you going to do about it? What corrective actions are you going to take? Is it reprocessing or disposing of the product, or changes in the process to make sure it doesn't happen again? There's got to be some type of corrective action to make sure that the preventative controls are working and that you don't have a breakdown again. And then finally, as part of the monitoring that you're going to do, you need to have some way of being able to verify those monitoring activities or testing activities. So there needs to be a verification process, and verifying that the testing is giving correct values and not giving you values that are incorrect that could lead to potential problems. So that's really what FSMA is all about. It's about creating a system where you have preventative controls in place, you've identified your greatest risks, you monitor that, you prevent problems from happening, and if you do see breakdowns, you take corrective actions immediately to prevent that from happening again. And there's obviously lots more incorporated into that in terms of the particulars of FSMA, but at its core, that's really what its purpose is. And so where does water activity fit in that concept, or that new process that is incorporated into FSMA? And really, if we think about it, water activity has sort of been waiting for this to happen all along. It's been a key part of the food code for quite some time now. And where it becomes a key part of it is that it was used as a way to define whether or not food could be determined potentially hazardous. In other words, is it going to potentially make somebody sick? But the key word there is potentially, because the way that water activity has been used up to this point within the food code has been as a preventative control. Its purpose was to prevent growth, not to eliminate growth or to kill organisms. Water activity is only preventative. It is not reactive. You can't have a contamination event on a food product that's happened and then use water activity to eliminate that. Because water activity is not a kill process. It only prevents growth from happening. So it is truly a preventative control. Its whole purpose within a food safety program has always been to prevent microbial spoilage, prevent things from happening that could potentially cause problems for the product. And really, if you've come to any of our virtual seminars, or if you haven't, I would encourage you to go to our website to watch those. And what we've always talked about is what can you use water activity to control? What can you prevent by having an understanding of water activity? And this goes beyond just food safety. As part of FSMA and it being a food safety program, we're focused in this discussion on how water activity helps to keep food safe. But beyond food safety and talking in terms of microbial growth, water activity is also critical in quality and maintaining shelf life of a product, whether that's due to microbial spoilage or it's due to other factors like chemical breakdown or changes in texture. And so we explore all of that in our seminars, talking about how to use this water activity tool as a preventative tool to prevent bad things from happening and extend shelf life. And that's really where its power comes in. And so it really is the epitome of a preventative control. It's really what its purpose has always been, and it's kind of been waiting for the system to catch up. And now we have here a system that's built upon risk and prevention that makes water activity perfectly set up to be able to play a critical role going forward in food safety programs that are established through the new FSMA regulations. And one nice thing about water activity is that thinking back in terms of what is the role of preventative controls, they have to be monitored and they have to be verified. And water activity is a very simple test to run. It's about a 5-minute test. There's instrumentation, very good instrumentation that we provide that does that testing. So there's really no if water activity if you're able to use that as one of your preventative controls, it's a fairly straightforward process to start implementing it and using it as a preventative control, to start taking and monitoring it using water activity testing equipment. And then you can easily verify that equipment through the use of salt solutions. So it really is set up to be able to be easily implemented. So if you've been using it, hopefully your fears, if you've had any that's going to change, have been taken away, because basically you're going to continue to use water activity the way you always have. If you haven't been using water activity, this may be the situation where you need to start implementing it to be able to meet these requirements for the new FSMA regulations. Especially if you've been using moisture content in the past. Moisture content is not a preventative control that meets the requirements of FSMA. And so if you've been using moisture content, certainly now is the time to switch over to water activity as an effective preventative control. And you can go in and look in the documents being created by FSMA, and sure enough, you'll see water activity as a critical part of that in discussing ways that or potential preventative controls. And so where can you actually find that information? So in the past, we saw water activity. It's been a part of government regulations for many, many years. You can find it in the Code of Federal Regulations for food production. It's been part of the definition for potentially hazardous foods for many years now, and even establishing whether or not your food could be sold off the shelf. But now as we move into the Food Safety Modernization Act and moving to a risk-based approach, again, that's similar to the HACCP programs. They're called HARPSI programs in terms of the FSMA, but they're very similar. So if it's something you're familiar with, it should be a fairly straightforward transition over to meeting FSMA requirements. Some specific places where you can find water activity as of right now, and keep in mind that there are still lots of documents coming out as part of FSMA. We're still going to see additional chapters coming out and additional information coming out that will continue to become available. But as of right now, and many of these, again, are in draft form as well, so be aware of that. But you can find water activity as a process control and a preventative control. Shown there, the reference for the chapter where that is. And you also start to see these new forms that are coming out, where you basically, the purpose of the form is for you to fill it out as part of your food safety program or to establish your food safety program. And so in that form, it asks you identify ways that you're going to, or what you're going to use as a preventative control or to make sure that your product is safe. What water activity levels are you going to target? Even wants to know, how are you going to control the water activity? Is it going to be through dehydration, or are you going to use humectants, and what kind of humectants are you going to use? And so there's some pretty specific information that's asked for there. I'm just have become acquainted with these forms. These are not ones that I had seen before. I don't know, you may already know about them and have been using them, but it's important to know that these are part of what is going to be required for you. Especially if you make intermediate moisture foods that are of concern for microbial spoilage. Now, water activity is also part of other systems. USDA has used a risk-based approach and a HACCP-focused approach for many years now. Pharma is adopting this as well to look for risk-based approaches for looking at whether or not your product is going to be potentially hazardous. So what is the mode of action then? How does water activity prevent growth? So if we consider an organism, a microorganism, basically what it is a membrane surrounding a sac of solute, or the interior has a specific water activity. And so when this organism, how this organism actually gets water is that it relies on movement of water from its environment into its interior so that it can go through its metabolic processes. When it encounters a water activity in an environment with a water activity less than its internal water activity, water actually moves out of the organism instead of in. And that causes a loss in turgor pressure. And that maintaining turgor pressure is critical to organisms to be able to continue their metabolic processes. And so the organism has to respond to this osmotic stress. And so it tries to lower its internal water activity. And it can do this through concentration of different solutes. And often, or each organism has a unique ability to be able to do this. And so each organism can get to a certain water activity. That's why they all have different growth limits. But as you in this example, it's able to get down to 9.3, but not down to 9.0. And so now it can't access water from its environment. And so it's forced to go into a dormancy stage because it can't go through its metabolic processes. So it's no longer actively growing. It's simply sits there dormant for quite some, and it can stay dormant for quite some time, but it's not actively growing to a point where it could cause an infection. And so we see that illustrated here on growth curves where we have log reductions or log growths based upon water activity. But this again is growth. We aren't killing, we're not reducing the organisms, we're simply preventing them from growing. So on this growth curve, we see that at high water activities, there's a very substantial growth curve. The initial stage is very short, and the stationary stage ends up at levels high enough, if we again consider a logarithmic scale, that cause an infection. But as you lower water activity, you cause a longer initial stage. The growth stage is being reduced, its slope is reduced, and so it isn't able to get to high enough levels to cause an infection. And this is due to the metabolic processes being slowed down and also having to readjust to try and concentrate solutes on the interior of the organism. And so from all of this, we can actually create these nice tables where we identify exactly where organisms will stop growing. So here are the pathogenic bacteria. Again, each organism has a unique value where it will stop growing based upon its ability to adjust or adapt to osmotic stress. And we can see the growth for molds. Again, these can actually grow at a little bit lower water activity, so they have a better ability to adapt. But only one organism will grow below 0.7, and it's not a typical spoilage organism. And that's why we call 0.7 the practical limit for microbial growth. So if you can get below 0.7, your product is safe from a microbial standpoint. And molds really aren't considered as potentially hazardous, aren't a critical part of food safety necessarily, because they don't get you as sick as pathogenic bacteria, as well as you can actually see the organism. And similar with yeasts, but the yeast, they stop growing at a higher water activity. There is an organism that can grow below 0.7, but again, not a typical spoilage organism. So again, if you're below 0.7 from a microbial standpoint, you should be in pretty good shape. But water activity, again, is not a kill step. Organisms can survive very well at very low water activities. Salmonella and staph can survive down to 0.26. And this wouldn't be a problem if you make an ingredient as long as your ingredient stays in that form. But if it gets added as an ingredient to a mixture where the water activity gets high, the problem here is that these organisms now become viable and are happy as can be to start growing. So water activity is not a kill step, it's a preventative step, just like we talked about earlier. And in fact, water activity actually is used in combination with other factors that contribute to microbial growth. And microorganisms need a lot of things to be optimal in order for them to be able to grow. And so putting different hurdles in front of the organism, they can be used synergistically to prevent growth. For example, in number 3 on the slide, you see water activity is of enough a hurdle to prevent growth. But in number 6, it's not. But when combined with the effects of pH, it does become an effective control. And so that leads us to these tables. These are part of the old food code in identifying or establishing a potentially hazardous food, but they're still applicable because they really feed right into this idea of risk analysis and preventative controls. So these tables are very useful in terms of telling you and helping you with your hazard analysis and risk assessment based upon what your water activity is and what your pH is. So if your pH, if you process your product with a kill step, so you're removing vegetative cells and then you package, then you meet the requirements for this table. And so you can be at 4.6 or less pH, you're good, no matter your water activity. Or if your water activity is at 9.2 or less, you're good, no matter, regardless of your pH. But you can even be at higher values as long as you have a combined effect. And so you're even at 9.3, if your pH is below 5.6, your risk analysis would be that you are not at a risk for growth based upon these tables. So these still become very useful. Keeping in mind that this is when the product is processed with the kill step and is still in the package. The minute that it comes out of the package, this doesn't apply anymore because now it's been exposed. But as long as it's in the package, then these values are applicable in terms of your risk. If it comes out of the package, or if you don't have a kill step, then you go to table B for your risk assessment, where if your pH is less than 4.2, doesn't matter your water activity, you're not at risk. Or if your water activity is less than 0.88, doesn't matter your pH, you're not at risk. But again, you can be at higher water activities and higher pH as long as there's a combined effect of the two. And so then you would use these tables again for your risk analysis based upon what are the properties of your particular product. What is the pH? What is the water activity? So hopefully in our short 20 minutes here, we've been able to introduce some concepts of FSMA, looked at what water activity's role will be in that, and hopefully have given you some taken away some stress if you were worried about, can I continue to use the water activity testing that I've been doing? The answer to that, of course, is yes. And the rules still apply. The same water activity values still apply. All of that is still true. It's just being applied as in food safety program as opposed to being applied just as part of the food code. And so water activity, again, it's designed specifically for this type of system. It's a preventative control. It's a critical part of what you'll do for your hazard analysis and looking at your risk assessments. Having that information in hand about what is the water activity of your product will be critical. And of course, we're here to be able to assist you in being able to figure that out. So please, if you have more questions, if you're interested in finding out more about the instrumentation for measuring water activity, we would encourage you to contact us. You can see our website listed there, as well as my email and our phone number. So feel free to please reach out and contact us with any questions. In the meantime, we do have some time now for question and answers. As was stated earlier, please submit your questions using that question tab, and you can begin to submit those questions now. And I'll be happy to answer any of those. If you don't feel comfortable asking a question now, or if you would prefer to ask your question one-on-one, then you can email me that question, or you can call me directly to ask that question as well. So with that, we will begin taking any questions that you might have. So one question that may be on people's mind is, does the water activity apply in terms of the numbers exactly the same way, and what kind of numbers are showing up in the documentation? If you go to chapter 4 that I referenced in the slides, you will see a discussion there about different water activities for different food values. They actually try to group them based upon what the expected water activity is. One word of caution: those tables are great from an informational standpoint, but we always encourage, and it's really necessary for you to actually measure the water activity of your product and do it routinely, because with that information, it gives you the ability to be able to do corrective actions right away, and be able to adjust processing if necessary, or take a look at potential problems that have come from ingredients. So those tables are there for information. We have additional tables. It's a great place to maybe get a start or an idea of where you're looking. But in the end, you really have to monitor water activity to be really doing what you need to as part of the FSMA regulation. So while we encourage you to then take a look at the instrumentation, it's not hard test to run. It's not an expensive test to run. So really, it's important to be able to understand how to effectively monitor water activity as part of your preventative control. Okay, we have some other questions coming in. One question is, can you use water activity readings in place of microtesting? And the answer to that is yes, as long as the purpose of your microtesting is to verify that you won't have any kind of microbial growth on your product. Now, if the purpose of the microtest is to test whether or not your processing is resulting in there being a contamination of your product, water activity isn't going to tell you that. It's going to tell you that it won't continue to grow, but it's not going to tell you if you've had problems with your manufacturing practices. So if the purpose is to figure out, are my manufacturing processes effective, or my sanitation processes, for example, effective at removing organisms, you'd still have to do some sort of a ATP test, or maybe another type of microtest to be able to identify that. But in terms of, and especially if you're dealing with pharmaceutical products, the challenge testing that has been required in the past can be eliminated based on water activity, because water activity is as a preventative control, determines or tells you as an intrinsic property that the organism can't support growth. So as long as you've verified your good manufacturing practices, then going forward, then there's no reason you would still have to do microbial challenge testing, because the water activity shows that there will be no growth. So there was a question that came in that is water activity as a preventative measure. If the number is not within the values that you establish, how can you correct it? Can you add, if you can't add solutes like salt or oil, because it might change the formulation? So which is a great question, and the idea of how can you effectively control your water activity to the value that you need to have? And really, that is a complex question. There is it typically takes a combination of efforts, but certainly one thing that will control water activity is your processing controls, both your time and your temperature. So how long is it exposed to certain temperatures, how long is or what temperatures is it exposed to will influence what the water activity will be of that product. So that's one way, is to adjust your process controls. That's not always a great option, and especially if you have to extend those, that can be problematic. But there again is where the value lies in being able to track water activity, because you don't want to make excessive changes if not necessary. But certainly, if your water activities are falling outside your range, and sometimes it's just a matter of establishing the correct water activity range, because we've seen lots of situations where folks have put in specifications for water activity that really aren't necessary. They may be overly strict, or they even on the other side are not strict enough. They could actually have the potential for causing problems. And this is especially true if you're trying to indirectly do it through moisture content. That's just you set yourself up for a world of problems. But even within setting your specifications for water activity, it could be that you may have set those too stringent, in which case you may find out that based upon the real risk, that you could widen those specifications or adjust those specifications. And then finally, to the idea, can you do it with solutes? And it's true that if you add solutes, you're changing the formulation, which could result in changes, undesirable changes. But there's also, and we've seen lots of successes using solutes, and especially if you can identify and use humectants that don't have a lot of bring a lot of impact um, on the overall qualities of the formulation. Things like, say, glycerol, which tends to be a fairly inert. It can cause bitterness at high concentrations, but there may be the potential to have some natural, even humectants that you could add that don't have a huge impact on the quality of the formulation, but will reduce water activity. And we have tools that you can use to actually predict and figure out how much of that you would have to add in order to change the water activity. There are tools available for doing that to try and help with that process. But in the end, it may be a combination of all of those that you need to use. And again, we're here to help, and we have systems beyond just water activity, but even systems where for data control and a collection where it allows you to do real-time determination of your data. It's a system called the Scala system, where the data is collected and you can see it on your screen real time, and use that to be able to identify times when corrective actions are needed, and see that right away, not wait for written records to come in, but have a real-time access to your data. So great question, and thank you very much. The question is, if the product is baked, what is the best temperature to test at for water activity? And typically what we recommend is that you measure the water activity, first of all, at the same temperature at all times so that you're consistent, but also under the conditions under which it will normally be stored, or in the temperature at which it will go to the consumer. And so typically those are done at 25°C, right around room temperature, as a standardization. You can read them at higher temperatures. One reason you might want to do that is if you're concerned about temperature abuse. And so you want to know, because water activity does go up with temperature. And so if you want to be able to get an idea of how much it might go up due to temperature, then possibly you could read it at higher temperatures. And some of our water activity instruments will control to higher temperatures. In a lot of cases, though, you can actually do a prediction about how much water activity will change with temperature by doing just a simple little study where you measure the water activity at 3 different temperatures, and then you can predict what the water activity will be at any temperature. And so instead of having to read at that temperature every time, you read at 25°C with an understanding that you could potentially get to a high water activity if the temperature goes up, and then you adjust your specifications based upon what could potentially happen. So to make sure that your water activity monitored at 25°C is always going to be safe. The other advantage sometimes to reading at higher temperatures, especially when you do have a baked product, is that you can sometimes test faster because you don't have to wait for the sample to cool down completely to room temperature. But again, if you're going to use that strategy, you just have to make sure you understand you're reading water activity at an elevated temperature, not at 25 C or at room temperature. But that is a possibility, and certainly possible with some of our instrumentation. Several questions have been about whether there will be slides made available. I'm happy to share a PDF of the slides with you. If you want to just send me a quick email, I'd be happy to send a PDF of the presentation to you. Okay, I think that's all of our time. I want to thank everybody for joining us today for this virtual seminar. I hope it was useful to you. I think we may be sending a survey to you about the usefulness of the virtual seminar, and particularly we'd like to know whether or not you liked the sort of shorter format to a virtual seminar. We don't want to have take up your whole day. We know that you have lots of things to do, so we're experimenting with some shorter virtual seminars. So if you have any feedback about that, we would love it to receive that. You can send the feedback in the questions, or you can email us directly and let us know what you think. So thank you again for your time here today and for your attendance, and we hope to be able to help you in the future, and have a great day.